So good afternoon. There's some people here in the room as well as over Zoom. We're here for our monthly rounds, mental health and addiction rounds. It's my pleasure to welcome today Dr. Annie Ciernia, who is an assistant professor in the UBC Department of Biochemistry and Molecular Biology. And her background is in neurobiology and behavioral neurosciences, as well as epigenetics and neurodevelopmental disorders. And today she is going to present microbes to microglia, therapeutic strategies for dampening neuroinflammation. So please welcome Dr. Annie Ciernia.
Thank you. I am super happy to be here today. Hopefully the people on Zoom can hear me okay. And I'm really excited to share some of the work that my lab has been doing. As most of you probably know, my lab is here on the Point Grey campus. We're actually right upstairs of the room where we're in right now. And this is the traditional ancestral and unceded lands of the Musqueam. And it really is my privilege to be able to do our science here on their traditional lands. And so the work I'm going to tell you about today is largely a collaboration that my lab has been doing with Carolina Tropini's lab, who is a microbiologist here at UBC. And the work that we've been doing really wouldn't be possible without people on sort of both ends of this gut-brain axis. And so what is it that I'm talking about? So there are literally trillions of microbes that live in your gut, and these microbes can control many host functions, and they actually can communicate with the central nervous system. And this can happen in a number of different modalities. So there's direct nervous system interactions between the gut and the brain. So through the vagal nerve, which is bidirectional actual connectivity between these two organs. There's immune cell trafficking. So immune cells can move back and forth between the gut and the rest of the body and the brain. And then there's a whole host of these rather interesting molecules that are made by the gut microbiome that can enter into the bloodstream and then either signal across the blood-brain barrier or travel across the blood-brain barrier and interact with cells of the brain. And one of the main cell types that is extremely sensitive to these metabolites from the microbiome is microglia. And these are the innate immune cells of the brain. If you've never heard of these cells, you're in for a real treat today. So these are mouse microglia that are tiled across the hippocampus. And you can see each one of these golden star structures is an individual microglial cell. And each one has its own territory that it is important for patrolling, and it's looking for signs of disease or injury. So these are a primary immune cell. They are responsible for fighting off infection, getting rid of dead or dying cells. But what's become apparent in the last 20 years or so is that microglia are incredibly active even in the homeostatic brain. And so this is some of the first in vivo recordings of microglia that was done now 20 years ago. And this is sped up a little bit, but you can see just how dynamic these cells are. They're constantly putting their processes out. They're interacting with other cell types in the brain. They're constantly sampling the local brain environment. And part of their function is actually to control the interactions of other brain cell types in early development. And so what's become clear in the last decade or so is that microglia play an important role in normal brain development. And they do this in part by helping to sculpt neuronal circuit formation across key developmental windows in early life. And one of the reasons we know this is because if you disrupt microglial function during some of these key time points in early development, you actually can disrupt normal neural circuit function and formation that ultimately leads to changes in brain development and behavior. And one of the key signaling molecules that is important for how microglia develop actually comes from the gut microbiome. And so some of these microbiota-derived metabolites that gut microbes can make can actually go into the brain and help signal microglia during early life to help them develop properly. And the reason we know this is that if you look in animals that are lacking these microbes, these are germ-free animals, they live in these giant kind of bubble units, they have zero bacteria or viruses or any fungi, their microglia develop abnormally. And so they have too many branches. Instead of having their nice own territories, they start to touch each other and they're hypo-responsive. So in an immune stimulating environment, like a viral infection, these microglia actually fail to respond properly. And this really suggests that you need these signals from the gut microbiome in order to form proper functional microglia in the brain. And so this is sort of the extreme condition. So you have normal microbiota, no microbes. And one thing my lab wanted to try to understand is what actually happens in a more physiologically relevant state. So in the context of something like gut inflammation, which disrupts the microbiome, does that also lead to changes in microglia function that may ultimately drive changes in behavior? And of course, what are the actual signals that are important coming from the gut microbiome to the brain? And so the model system that we decided to explore was actually a model of inflammatory bowel disease. And so in the context of this disease, which is well characterized in humans and in animal models, there is an increase in gut inflammation and a corresponding change in the gut microbiome. And there are depletion in the diversity of microbes that are present in patients with IBD, a decrease in the circulating metabolites that are then released by these microbes. And importantly for us, there's at least some data to hint at the fact that patients with IBD may have changes in their brain. And so IBD patients have a increased prevalence of anxiety and depression. They're also at higher risk for age-related cognitive impairment, including onset of dementia and Alzheimer's disease. They also have changes in white matter structures in the brains on MRI. However, these are people living with a chronic disorder, and so it's really hard to draw a cause or consequence from this type of human data. However, there are a number of animal models which also are able to recapitulate some of these brain changes and the anxiety and depression phenotypes. And so this led to our hypothesis that maybe if we could model this gut inflammation in IBD in young animals, we could start to get an idea about how that might alter brain development, potentially leading to lifelong changes in behavior and the developing brain. And so to do this, we developed a model to look at early life gut inflammation. And we did that using dextrin sodium sulfate treatment, which you can just give in the water. So DSS is a collagen. It disrupts the mucus lining of the gut, and this causes a gut irritation, which leads to inflammation and changes in the underlying microbiome. At least that's what happens in adult animals. So we had to test this out in young animals. So we did this starting at weaning. The young mice get 5 days of DSS treatment. They get 2 weeks of water to recover, another 5 days of DSS during adolescence, a couple of weeks to recover, and then another 5 days of DSS in young adulthood. And we did this repeated paradigm to mimic the relapsing and remitting components of what kids with IBD often have. So they'll get flares, followed by recovery, followed by flares. And so we wanted to hit at least a couple of key developmental windows to see what might actually be the impact of changes in the gut microbiome during this time frame. And so the first thing we needed to do was to actually see what happens to the gut itself. And so this was work done by Claire and Carolina's lab to actually look at the gut tissue. And so what's shown here is the colon. So you can see the DAPI labeled blue nuclei of the endothelial cells, the green mucus layer and the red bacteria, and the control animals. Those are all nicely separated. In the DSS treated animals, that mucus lining is disrupted exactly as you would expect from treatment with a collagen. That's what it's supposed to do. And we have changes in the bacteria interacting with the host. So that was a good sign that we were able to induce gut inflammation in these animals. Then Olivia Sullivan, who's my master's student at the time, and Cal Rosette, who was an undergrad in the lab, went ahead and did a heroic amount of behavior on these animals. We looked at anxiety, depression-like behaviors, repetitive behaviors, cognitive performance, social behaviors, the whole gamut. We ran 3 complete independent cohorts of animals. And unfortunately for Cal and Olivia, this was a bit depressing at the time. There was actually no changes in any of these measures. And this was a bit unexpected because adult animals, when you do this DSS treatment, often have anxiety and depression-like behaviors. But when we went back to the literature and looked at this more closely, many of those tests are done very close to the DSS treatment. So you're potentially also dealing with some sickness behaviors that might confound this. Our animals were completely recovered by the time we do these behaviors. And so there appears to be no long-lasting changes that we could detect when you do these early life changes in the gut inflammation. However, one of the things that we did find, which was unexpected, is that when we were collecting organs from these animals, we noticed that the male animals had significantly smaller seminal vesicles. And when we actually did this, I actually thought we got the sex of the animals wrong. So the seminal vesicles were smaller and they were actually tucked back up inside, indicating that they were underdeveloped. My postdoc Jordan, in collaboration with Kieran Soma's lab, then went ahead and looked at some of the circulating sex hormones in these animals and found that both testosterone and androstenedione, which is the precursor to testosterone, were significantly repressed in the male animals that received DSS treatment. And so this was sort of our first indication that we had some sort of neuroendocrine function that we had disrupted with our inflammation model. And so Olivia and Cal went back to the drawing board, and this was really driven by Cal, who did this as her part of her honors thesis. And so we developed a set of tests to look at more sex-specific behaviors in these animals, specifically in the males. And so I'm just going to show you the results of the urine preference test today. And this was what Cal and Olivia came up with. So male rodents will typically seek out female partners using olfactory cues from the urine. And so we gave our animals a choice. They could explore either female urine or water, and then we just asked them where they choose to spend their time. And so the vehicle-treated animals show a significant preference for the female urine. The DSS-treated animals do not. If anything, it's actually aversive. And importantly, we did check the olfaction of these animals, so they can actually smell just fine. They can discriminate a social versus a non-social odor. They can tell two females apart, but they don't seem to care about interacting with the female urine, indicating that there's some kind of mate-seeking deficit or some kind of rewarding behavior of the urine that they are no longer experiencing. And so at this point, we had now some indication that we'd changed sort of sex organ development, changes in testosterone, and changes in sex-specific behaviors. So we went back to think about how this might be related to the microbiome. And it turns out there's actually a known pathway by which the microbiome can regulate circulating levels of sex hormones. And so sex hormones in the blood, both estrogen and testosterone, they're on modified form. When they're to be excreted from the body, they go to the liver, where they have a chemical modification. So a glucuronic acid is added to them that then causes those molecules to be excreted out in the urine. And typically that would then result in them leaving the body. However, in the gut, there are enzymes that express a GUS enzyme that can remove that glucuronidate, and then the deglucuronidated form can exit back out into the blood. So the net result of this is if you have high levels of these GUS producing bacteria, you can increase the circulating levels of estrogen and testosterone. And so Claire wanted to take a look at our animals and see what actually was happening to changes in the microbiome. And so both the males and females have decreased diversity of the microbiome. And you can do this by a couple of different metrics. These are just the two that I'm showing here, but you can see that both the vehicle male and female animals nicely separate from the DSS treated animals. And we see a decrease in the diversity of the microbes present in the DSS treated mice. When we look specifically at the bacteria that produce this GUS enzyme, their levels are actually positively correlated with circulating levels of testosterone and androstenedione. So the higher you have of these GUS producing bacteria, the higher the circulating levels of basically male sex hormones in the male animals. However, that doesn't really tell you about the function of these particular enzymes. So Claire took the cecal contents from these males and actually measured the enzymatic activity of the GUS enzyme, and that is also significantly lower in the DSS-treated males, indicating that this is potentially one mechanism by which the males are failing to convert androgens back to their active form, and this might explain the decreased levels of testosterone and androstenedione in these animals. At this point, we wanted then to try to connect this back to the brain, and so my PhD student, Jennifer Kim, went ahead to actually look to see what happens to the microglia in the brains of these animals. And to do that, she used a new tool that our lab had been working on. So microglia can actually take a number of different cellular morphologies. So I showed you the video with the nice ramified microglia that have all these processes that are reaching out and touching other cells. But under different types of brain conditions, you can see different prevalencies of other types of shapes. So under inflammatory conditions, microglia retract their processes and form these amoeboid cells, which are thought to be pro-inflammatory. Under stress-like conditions, you can see an increase in hypertrophic microglia, which are about midway between amoeboid and ramified. So they're smaller, more compact, but they still have some branches. And then around blood vessels, and in the case of brain injury, you can often see an increase in these rod-like microglia, which are thought to interact with the brain vasculature. And so Jen made a tool to go ahead and quantify the proportion of microglia in each one of these different cellular morphologies. And to do that, how this works is we do brain sectioning, stain for your favorite microglial marker, and then parse out individual microglial cells. We can then analyze 27 different metrics about their cellular morphology. So like how branched they are, how much space they take up, how long each branch is. We then take those 27 metrics and do some fancy dimensionality reduction. And then we take those principal components and cluster them. The end result of this is that you can identify the proportion of microglia that make up each one of these individual states that is completely independent of what each individual metric might be doing. And if you're interested in this, this is now published and the code is all freely available. Jen also made a very nice YouTube tutorial if you want to watch about how to make microglia morphologies, and you can play around with some of your own images or some of ours. And this was specifically designed to be able to be run by undergrads in the lab. And so this is meant to be as user-friendly as possible. Okay, so what did Jen actually find? So when we went back and looked at our DSS-treated animals, we took a look at the microglia and we were able to use Jen's tool to identify the proportion of microglia in each one of these different states. When we look at the female mice, we can look at the proportion of microglia in the amoeboid, the rod-like, the ramified, or the hypertrophic state. And what we see is a significant decrease in the number of ramified microglia in the females and a trend towards an increase in amoeboid microglia. And this is consistent with a pro-inflammatory sort of state in the brain of these animals. When we look at the males, we see something that's actually a bit different. So in this case, we see only an increase in hypertrophic microglia. And hypertrophic microglia are often seen in the context of chronic stress models. They're also seen in some neurodegenerative disorders. So this appears to be something that's also not a homeostatic state, but something that's a bit different than what we see in the females. And we don't yet know what these different morphologies mean in terms of driving changes in behavior. But I think it nicely emphasizes why it's important to look at both sexes in your experiments, because you might get something completely different. And so at this point, I was presenting some of this data at a conference, and I met this PI from Wisconsin, JPU, who has been developing ways to try to image microglia in humans. And he does this using sort of an indirect measure of diffusion MRI. And so water diffusion is actually sensitive to the underlying tissue microstructure. And you can use this to your advantage to get information about what the inflammatory state of the tissue might be. And so if you see that little red bouncy ball at the bottom, and under homeostatic conditions, it has a lot of degrees of movement. Under inflammatory conditions where you get proliferation of microglia or changes in morphology, that water diffusion movement is restricted. And JP is able to measure that change in the water molecule diffusion in his MRI. And this he refers to as the neurite orientation, dispersion, and density imaging, also called NODI or ODI for short. And this is sort of a surrogate measure of microglia density. And the reason that he knows this is sensitive to microglia is that he performed this rather clever experiment where he gave mice a Plexicon drug, which is an inhibitor of the CF1R receptor that microglia need to survive. So he basically killed off all the microglia in the brain and then took the animals off the drug, and the microglia slowly repopulate. And he collected brains at multiple time points. So he has a controlled setting where he has different levels of microglia in the brain. He then compared this to his ODI signal and saw a nice correlation between the density of the microglial cells and aspects of this ODI signal, indicating that the ODI signal is sensitive to changes in the underlying tissue microstructure, especially in the context where you're manipulating microglia. And this is quite exciting because you can actually do this in humans. And so this is one of the first attempts to do so. And this was, instead of done within individual humans, it's done across. And so what they did was took a bunch of healthy participants and measured this ODI signal. And so you get differences across brain regions. And those differences in ODI signal actually correlate quite nicely with known differences in microglial density in different brain regions from postmortem human brain samples. And so what this suggests is that you could use this ODI measure potentially in the clinic to look at sort of changes in response to any kind of manipulation to microglia that you might be interested in. Okay, so if we go back to the actual experiment, what did JP and his lab find? So what they found was that we had an increase in the ODI signal in both males and females in the hippocampus. And this is the same brain region that I showed you the differences in the microglial morphology in. And interestingly enough, he did not see differences in other brain regions. So we looked at frontal cortex, thalamus, hypothalamus, and we looked at these regions also for changes in microglia and did not see anything there. So this is pretty consistent across different measures. Why the hippocampus is so sensitive, I don't have a good explanation for, but it appears to care a lot about these signals from the gut microbiome or the inflammation that is being driven by this model. And so at this point, we now have this nice model in which we have demonstrated changes not only in microglia, but also the underlying metabolites and the microbiome, and that how in males leads to changes in behavior and neuroendocrine signaling. At this point, we decided to explore what some of the other metabolites might be that are actually signaling to microglia and how these might be changing across early development. And so there are actually a ton of these different microbe-derived metabolites that can be signaling to the brain through the blood. I'm just going to show you some work that my former master's student Vivian did on one category of these called short-chain fatty acids. So short-chain fatty acids are actually derived from the diet. So when you eat dietary fiber like fruits and vegetables, they are broken down by microbes in your gut to make short-chain fatty acids. And these are acetate, propionate, and butyrate. All three of these are fairly small molecules. They can diffuse into the bloodstream and diffuse across the blood-brain barrier and interact directly with cells inside the brain, including microglia. And so we know that these can potentially get into the brain. And we know that they're important for microglial development because if you give animals that have no microbiome back some of these short-chain fatty acids, you can improve some of the developmental impacts of missing a microbiome. So they're able to rescue some of the changes in microglia. However, it's not really known how these actually function in a normal condition or under inflammatory conditions, although they're thought to be potentially anti-inflammatory. And so we decided to go ahead and investigate what short-chain fatty acids might do in the context of brain inflammation. And so during inflammation, microglia undergo a number of different processes. So they increase nitric oxide production, they make pro and anti-inflammatory cytokines, they start to phagocytose dead or dying cells, and they actually will proliferate. And so this gives us a whole basket full of different measures that we can do in order to understand how microglia might be responding to different inflammatory stimuli and how short-chain fatty acids might impact that. All of these processes are intricately linked to changes in gene expression. And so one of the main mechanisms that has been implicated in how short-chain fatty acids might control inflammation is through modulation of histone acetylation. And that is the addition of post-translational modifications to the histone tails. This changes how they interact with the underlying DNA. And the more acetylated your histones are, the more it tends to open that structure and promote gene expression. And this is controlled by two classes of enzymes. So there's histone acetyltransferases that add these acetyl groups and deacetylases that remove them. And so it's been proposed in the literature that short-chain fatty acids may act as an inhibitor of the HDAC component of this, which would be predicted to drive the reaction in the favor of HATs towards increased gene expression and more expression of these pro-inflammatory or whatever kind of genes are going on at the moment. However, this is a bit confusing because short-chain fatty acids are thought to be anti-inflammatory. So this sort of left us with the question, how does this actually work? And so to do this, we decided to set up an in vivo model in which we could look at microglial inflammation. And to do this, we wanted to start with something incredibly simple. And so we actually just dose our animals with lipopolysaccharides. So this is the outside of a gram-negative bacteria. So if you've ever had food poisoning, this would be one of the reasons that you don't feel so well, is that you're getting this LPS into your body. And when we dose mice, what we can see within a few hours is that the microglia go from this nice homeostatic state, we add LPS, and they become inflamed. So they start to produce pro-inflammatory cytokines, they change their morphology. And really, this gives us a nice way to control inflammation in the brain in a very time-locked and dose-dependent manner. And so we decided to use this as a way to induce inflammation and then to examine the impacts of short-chain fatty acids. And so what we decided to do was follow this treatment regimen that had been used in the literature, where we dosed animals with either vehicle, which is just salt water, butyrate, propionate, or acetate at different doses. And these doses are meant to mimic sort of the endogenous levels of short-chain fatty acids in the blood. We then do this for 4 weeks to let this accumulate, and then we give them a single injection of LPS or saline, and then look to see what happens to the microglia. And so this was work done by a whole bunch of people in my lab. This takes a lot of animal monitoring and a lot of work. But the first indication that these short-chain fatty acids might be modulating the LPS response came when we were monitoring the animals after the LPS injections. And what we found is that LPS nicely induces sickness behavior in these animals, and that was significantly blunted by the short-chain fatty acid treatment, especially the short-chain fatty acid butyrate, which was able to significantly blunt the sickness behavior in both males and females, almost back down to baseline. And so this was the first indication that, yes, short-chain fatty acids, at least globally, are probably doing some kind of suppression of sickness behavior or inflammation. We then wanted to look at, okay, what actually happens to the microglia? And so to do that, we decided to use a tool that my lab had developed to look at global changes in histone modifications. And we can do this within isolated microglia from the brain. And we can do this at the single cell level, and you can multiplex it. So we can look at a bunch of different histone marks within microglia, even a very small number of them. And this is done by flow cytometry. And so to do this, Morgan, my PhD student, went ahead and profiled a couple of histone marks. So we looked at histone 3 lysine 9 acetylation, and this is a acetylation mark that's often found at promoters of active genes. And what she saw was that LPS was able to increase this mark in the vehicle-treated animals, and that was actually expected. But what was a bit unexpected was that these acetylation marks were hyper-induced in the females when they had butyrate, propionate, or acetate in combination with the LPS. And so everything went up, and it went up way higher in these animals that had short-chain fatty acids. Interestingly, this did not happen in the males. And so in the male animals, they get an increase in histone acetylation at baseline just with butyrate and no bump up with LPS. And so this is also true for another histone mark. So when we looked at H3 lysine 27, which is an enhancer mark, when we look at acetylation of that location in the genome, what we see is actually very similar. So in females, we get an increase when they have short-chain fatty acids in the LPS. The males do not. They only have an increase in butyrate at baseline. And so this really indicates some kind of sex-specific response of short-chain fatty acids in combination with inflammation, and that this doesn't really seem to happen in the males for mechanisms that we don't yet completely understand. So at this point, we have some changes in what we think is an epigenetic regulator of gene expression. We then wanted to look at what actually happens to gene expression in these animals. And so we did RNA sequencing on isolated microglia from these same conditions. And what I'm showing you here is what happens just in the vehicle-treated animals with and without LPS. So this is a heat map where each row is a gene and each column is a sample. The samples under the sort of black column are the vehicle-treated animals, and the red column is LPS. And what you can see is we have a bunch of genes that just respond to LPS and microglia. So that was good. That's sort of the positive control. And these are genes that are important for the things that you would expect to be responsive to LPS, things like phagocytosis, nucleotide formation, lysosomes, proliferation, those sorts of things. When we look at what happens to the animals that are treated with butyrate before they get LPS, the story is actually a bit different. So in that case, these genes are all significantly blunted. And so it looks like the short chain fatty acids, and what I'm showing you here is butyrate, but this looks very similar for propionate and acetate. They're able to blunt the induction of LPS responsive genes in microglia. We also get a set of genes that are regulated uniquely under butyrate treatment conditions. And so these genes are not significant in our saline treated animals when you give them LPS, but they are significant in our animals that have butyrate. And so it enhances the LPS response in those animals. And many of these genes are actually ones that are known to be responsive to the gut microbiome in microglia. And so now this gives us a number of pathways we can start to look at and start to get an idea about how short chain fatty acids might actually be driving changes in the underlying microglia. So this is what happens for propionate, acetate, butyrate, acetate, and propionate look very similar. And so we decided to overlap the genes to see if there was some kind of conserved signature of how short chain fatty acids might be regulating microglial gene expression. And what we found is maybe not that surprising, but there's some overlap and some unique. And so if we look at genes that are increased or decreased in response to LPS that are blunted by short chain fatty acids, you can see in the center of these that we have genes that are similarly regulated in all three short chain fatty acid treatments, but then there's some that are not, many that are unique to butyrate or to propionate or acetate. And similarly, when we look at the genes that are regulated only under short chain fatty acid conditions. Again, we have some that are conserved, but now many of them are actually uniquely related by different short chain fatty acids. So even though these short chain fatty acids are chemically quite similar, they seem to have some non-overlapping impacts on how microglia are being regulated. We then decided to go ahead and actually take a look at what the microglia were doing. And so we used our morphology tool to look at changes in microglial morphology and a set of animals undergoing these same conditions. And this was work done by Cleanta in my lab, who's actually an undergraduate. And what she did was use Jen's tool to identify these 4 different microglial morphologies and then quantify the proportion of microglia in each one of these different cellular conditions. And what she found was that, especially in the female shown in this red box, you get an increase in amoeboid microglia with LPS, exactly like you would expect. And this is actually significantly blunted in all 3 of the short chain fatty acid conditions, indicating that in females, short chain fatty acids are able to blunt this transition to a more pro-inflammatory microglial state. Interestingly, this does not happen in the males. In the males, you get an increase in amoeboid microglia, and it's not sensitive to short chain fatty acids. You get the same amount of amoeboid microglia regardless of what short chain fatty acid treatment you have. Again, indicating that there's some sort of sex-specific effect for how short chain fatty acids are interacting during neuroinflammation in these animals. So at this point, we decided to do some more mechanistic studies of how microglia might actually be regulated in these contexts. To do that, we developed an in vitro model where we give, again, sodium butyrate, sodium acetate, or sodium propionate in the cell culture media. And in this case, it's in BB2 microglia, which are actually female. We then give the cells LPS and, again, wait for 24 hours to collect them and look to see what happens. So the first thing we did was to make sure that this model can recapitulate at least some of the things we see in vivo. And so when we look at changes in histone acetylation, again, we see a strong induction with butyrate, especially in the context of LPS, and also in this case in propionate. Very similar to what we see in vivo. When we look at changes in some of the genes we identified in vivo, again, we see an increase with butyrate and in the context of both saline and LPS. So this indicates that our in vitro model is able to capture at least some of the aspects of what we see in vivo. We then went ahead and used this model to look at microglial function. So we looked at nitric oxide production, which you can do just in a plate-based assay using the grease reagent. And what we found is that LPS is able to induce nitric oxide, so that's the blue bars, as you would expect, and that this nitric oxide production is actually blunted when you have treatment with any of the 3 short chain fatty acids, indicating that the short chain fatty acids are able to blunt this production in nitric oxide synthesis. We then looked at proliferation of microglia, and so we can do this by adding in BRDU, which is a thymidine analog. You can add it into the cell culture media. It gets incorporated into any dividing cells that we can then quantify using flow cytometry. And so when we look at the amount of microglial proliferation, we see that this is increased with LPS and significantly blunted with butyrate. It's not blunted, however, with propionate or acetate. So this is a butyrate-specific effect. But it looks like this is able to dampen down that increase in LPS-induced proliferation. And the last sort of functional measure we did was looking at microglial phagocytosis of E. coli. So microglia in the brain typically don't see E. coli unless you have some really major changes in the blood-brain barrier, but this is a good way to sort of measure general microglial activity in culture. So we give them E. coli tagged fluorescent beads, and these beads actually have a pH-sensitive fluorophore on them, so they only fluoresce when they're inside the phagolysosome, so they have to be engulfed by a microglia to be able to see them. And what we found is that both at baseline and in response to LPS, butyrate is significantly enhancing phagocytosis and actually propionate as well under baseline conditions. And this actually was a little bit perplexing. So far, what I've showed you is mostly changes that appear to be sort of suppressing inflammation, but in this case, we're actually enhancing microglia phagocytosis. And so I didn't show you this, but we're also able to enhance some cytokine production, including both pro and anti-inflammatory cytokines. We decrease nitric oxide, we're increasing phagocytosis, but decreasing proliferation. And so this is, at first glance, maybe a bit confusing, but what I think this really suggests is that instead of being anti-inflammatory, short-chain fatty acids, and particularly butyrate, are actually promoting the resolution of inflammation. And so you have to, in order to resolve inflammation, you have to sort of increase some of these properties like getting rid of dead or dying cells. You have to increase certain cytokines that can help promote the resolution, but you also want to turn down some of those things that actually promote sort of chronic inflammation like nitric oxide or proliferating cells. So this is still work that's ongoing in the lab. And of course, like most of science, this leaves us with more questions than answers. But what I've shown you today is that it looks like early life gut inflammation does not impact the vast majority of behavioral development, at least in the context of when we gave the DSS treatment. However, our work does suggest that especially in boys as they transition to young men, that this particular population may be especially vulnerable to suppression or delays in puberty onset. And this is consistent actually with case studies in boys with IBD that often have delayed puberty onset and with men who have reduced fertility. However, this excitingly does suggest that there may be alternative treatments besides hormone replacement therapy for these patients, including potentially probiotics that increase the production of these GUS-producing bacteria. We also found that in the second part of this, that the short-chain fatty acids are able to potentially promote resolution of brain inflammation through a mixture of sort of responses, including changes in inflammatory gene expression, proliferation, and nitric oxide production, and then also enhanced phagocytosis. So of course, this still leaves a bunch of work to do. We're starting to look at how butyrate might be able to reverse changes in our DSS model to microglia and maybe some of these hormone-related changes. We could potentially try to supplement in some of these GUS producing bacteria to see if we can also rescue changes in that model. And of course, we want to know how the short-chain fatty acids are actually causing these changes in microglia. There's some debate in the field about whether they have a specific receptor or if they're diffusing directly across the membrane. And of course, there's a whole ton of other metabolites we can start to explore. We've started to do some work on some secondary bile acids and some other components of gut-derived microbiome metabolites that we're excited to pursue further. And sort of one future direction that I'm excited about is we now have this model where we can use the human microbiota to drive potential changes in the brain. And so we can take our germ-free mice and give them human fecal samples, and you can use human donor samples to colonize them, and about 60% of the microbes will successfully transplant. We can then mate those animals and look at the offspring. So the offspring inherit their mother's microbiome. And so these animals will have the human microbiome throughout their entire life. And so this gives us a way to potentially look at how alterations to the microbiome can actually potentially have a causal contribution to changes in the brain and behavior across early development. So that's work that's currently ongoing in the lab. And of course, none of this would be possible without all the great people in both labs who did all of this work, both mine and Carolina. And of course, all of the funding that supported this work. And then the first part of this actually was published earlier this year. So you're welcome to check it out in BBI. And with that, I'm happy to take any questions.
The last thing you showed,
Mhm.
fascinating, about the fecal transplant and transfer from
Mhm.
one generation to this. Are there any other examples of that happening?
I mean, this is a pretty artificial setup. So these are the only microbes that are available to colonize them. In sort of the natural environment, we do tend to inherit our first microbiome from our mothers, especially if you have a vaginal birth, then most of the first colonization is from bacteria that are present in the vaginal canal. Or if it's born cesarean, they actually tend to be skin microbes that first colonize, although those differences tend to go away within a week or so. So it's a little unclear how much that actually really matters. They tend to change. Most of the early microbiome then is driven by milk production. So what you can get from either breast milk or formula, or in this case, from the mice. So a lot of it is bacteria that are used to process milk. That's basically what the majority of them are up until they start to eat solid food. So you can see major shifts in the microbiome of mice around P14 to 20 when they start to chomp away on some solid food, and then certainly after they're weaned from their mom. You get a big sort of shift in what's there. And the same for humans when they start to eat food. And then by the time kids are sort of midway through childhood, it's thought that your microbiome is fairly stable. There are exceptions to that. If you have like large dose antibiotics or something else that really disrupts things. But even then, like post antibiotics, usually you repopulate with sort of the same microbes that were present before. So enough survive that you sort of repopulate back with what you had. It's actually not trivial to change out the microbiome of an adult human. Like even with lots of antibiotics and probiotics, it's pretty hard to change things. And that's mostly because you evolve with your microbes. And so the ones you have evolved with you and are really well suited to the little niche that they have in your particular gut. And because microbes divide multiple times a day, they have basically thousands of like iterations of evolution across the human lifespan. So they are really well suited to an individual. And so it's very hard to replace them. However, I'm excited about, even if you can't replace all your microbes, probiotics are still useful when you're taking them because as those microbes move through your system, they're able to digest fibers and produce metabolites. They maybe don't colonize you, so they're not taking up permanent residence, but they can still have positive impacts by what they're doing metabolically. You just have to keep taking them. And of course, none of those microbes can do anything if you don't give them the proper food. So most of these probiotic type treatments, I think, would have to be sort of accompanied by some dietary guidelines as well. Yeah. Yeah.
In terms of, it was certainly for you, but a bit surprising for me for sure, the sex differences.
Mhm.
And in terms of animal models, do we have a different vulnerability in terms of what we can measure in animal models for sex? Like we have animal models that in a way follow the same trend of anxiety and depression models for sex, considering sex.
Yeah. So yes and no, I think the answer is. I mean, there are known sex differences in how mice and rats will respond to different behaviors and different stressors. So, for example, one of the common models in rodents of like depression is a chronic stress model. And males and females do have different vulnerabilities to that and will display different behaviors as a result. What drives that, I think, is still an open question. And whether it mimics exactly the same sex differences you would see in humans, I don't know if that's potentially the same mechanisms. But certainly there are differences that we know in the underlying biology that happen, especially in the context of microglia, that are not completely clear what drives them. But when people have done like transcriptional profiling or functional profiling of microglia across development, there are subtle differences just in healthy animals between sexes. And that seems to be amplified in the context of stress or a disease context or something else, to the point of where you can drive quite stark differences depending on what kind of manipulation you do. Again, why that happens, it's not completely clear. It's probably a combination of the sex chromosomes and the brain environment that's hormone driven or whatever. There's this cool experiment where somebody gave male animals one of these plexicon drugs and depleted all the microglia so they go away. Then they took microglia from a female adult mouse and just transplanted them in. And those microglia end up being kind of in between. And so they have some of the aspects that they maintain from their original like female environment that are probably driven by the endogenous cellular context, so some chromosomal thing or programming. But then they take on sort of some of the changes that you would expect from the local brain of the male animal. So it's probably some of both. But that's like the only good example I know where someone has attempted to try to sort those things out. It's a bit complicated. But yeah, I mean, my lab always runs both sexes in all of our experiments, mostly because we often see sex differences in the behavior or the responses to the microbiome. But often what they mean in the context of how you translate it to humans is not necessarily always clear. It looks like there's a question from online.
Yes, actually, I have two. First and foremost, actually, with this question or this slide that you have showing, are you looking at any level of engraftment of the healthy donor tissue into your mice? And if so, how are you doing that? And then my second question is actually related to your DSS and aversion to the urine. In that, I'm wondering, granted, I know it's probably very difficult to quantify, but if it's actually possible for the mice to feel like the DSS and the urine actually smell similar, and so it actually does create an aversion.
Yeah, okay. Those are both really great questions. I'll answer your first one first. So, in terms of engraftment, so yes, so we have human donor samples that are collected. And first of all, when you collect fecal samples from humans, not all the microbes will survive. So, things that are not tolerant to air typically don't survive well. It doesn't matter how well you collect them, it's pretty tricky. We do have some samples, and actually the ones used for this particular study that were actually taken during a biopsy. And so those ones do have the anaerobic microbes in them, and they were processed very carefully with the intent of trying to keep those alive, but it's never perfect. Once we put them into the mice, there are some microbes that just will not survive. Like they're too adapted to their human host and they just won't live. The way we actually quantify how well the engraftment worked is we can sequence the microbiome from the healthy donors. And then we can sequence the microbiome that comes from the moms, and then we sequence the microbiome that comes from the offspring. And then we can compare those. And so about 60% of the total like overall species that are represented in the human transplant decently into the mice. And then when we go from the mom to the offspring, they're almost identical. And now we've done actually a couple generations out, and they're fairly stable by that point. There's always some variation animal to animal, but once they engraft, they appear to adapt to that mouse host, and then they're fairly stable. Right now, we've only done one pair of healthy donor and one IBD donor. So we'll see how well that's maintained across multiple different samples collected from like different people at different times, but that's at least what we've seen so far. And your second question was about the DSS treatment, and remind me what the rest of it was. Oh, about the urine, of course. Yeah. So what I can tell you is that the urine is collected from a totally different set of just wild type female animals. So they have no inflammation or any changes at all. And it's the same urine. We actually pulled from a whole bunch of them and then aliquoted it out. So it's the same urine used across the entire experiment. And so that's held constant. Whether the DSS treated animals truly find it aversive, I don't know if we can really say from this. I think you need some more sophisticated testing. But they certainly don't like it. And they can smell females, and like we did test that they can discriminate one female from another or a male from a female. So it's not that they can't detect it, they just seem to not want to spend time with it. And yeah, and then what that means in terms of what a human would do, I don't know. Human men typically aren't olfactory driven to seek out males, but maybe there's something else about the rewarding aspects of a social interaction that might be different. Not sure. But and the reason I didn't show any of this data, but we didn't really see anything in the female DSS mice in terms of like sex-specific behaviors. But I largely think it's because doing those kinds of experiments in mice are just not a good model for that. Female rats will show this like stereotyped lordosis pose that's receptive to males. Female mice don't really do that. Their response to males is to try to hide. And so that's not really the most useful measure for looking at changes in mating behavior. And so I think the jury's still out on what is exactly happening in the females in this model. It's really difficult to measure estrogens in female mice. Their levels are incredibly low, which is very different than humans. In fact, we can't even pick it up with the most sensitive mass spec on a sort of consistent basis. So we're working on some more sensitive measures, but right now that is actually not technically possible. So again, I think rodents might just not be the best model for looking at the effects of inflammation on like female sex-specific behaviors.
Thank you so much.
Yeah. Yeah.
General question, I'm wondering if a person's microbiome is pretty stable over a lifetime, how does, diet or even like natural supplements, or for example, Tony and I are working on a psychoactive plant-based formulation that you take orally. It has things like ginger and cinnamon and things that, do things on the GI system, but then there's also psychoactive components and effects on dampening neuroinflammation. Like, how could that be related?
Yeah, so what the microbes you have are relatively stable, but the abundance of them, the relative abundances of them can shift actually quite dramatically with diet. So you're not going to like magically invent and get new microbes typically, but you can shift the composition of how much of different ones you have by what you feed them. So for example, if you eat a lot of high fiber foods, you will cause sort of a bloom in microbes that eat that as their main food source, which tend to be the ones that actually produce short-chain fatty acids, for example. There are, of course, limits to that in terms of you don't really want just short-chain fatty acid producing bacteria either. So having something like a more balanced diet probably is a good idea. But yeah, you can see actually, and some of this data actually comes from some interesting work that's done in indigenous tribes in Tanzania, where their diet is highly linked to, so they're hunter-gatherer sort of civilization still, and their diet is highly linked to the seasons. And so during one point in the spring, their diet shifts to like 80% honey because they have it available. So it's this massive dietary change, and you can see a huge corresponding shift in the microbiome. And then when the honey becomes unavailable, after however long, the microbiome shifts back to sort of its pre-stable state. So it's not like they have a whole new set of microbes. It's just that you get a bloom in those microbes when the right food sources are present. We typically, because we live in a Western society where food is fairly available, you can go buy blueberries if you want year round, don't have those huge dramatic seasonal shifts that you see. And so what that means for how our microbes are adjusted to that, I don't know, but it's certainly interesting to think about. Yeah.
Thank you.
Yeah, thanks for having me.